start with some other proteins of clinical interest. Ben-Jones protein, an abnormal protein, occur in the blood and urine of people suffering from multiple myeloma, plasma cell tumor. What is multiple myeloma? Plasma cell. It has a low molecular weight of 45,000. Dekho, you don't have to memorize this molecular weight. I only tell you this because I want to, you should know that the molecule is heavy or it is small or it is large or it is light. So just this tells you about approximate size of the molecule. It consists of light chains of immunoglobulins, either the kappa chains or the lambda chains. Don't get confused with this. When we'll go with the immunoglobulins, when we'll study the immunoglobulin lecture, so you should know, okay, you will come to know, okay, what are the kappa chains, what are the lambda chains. So whenever it consists of, so what we have captured, that it is secreted, it is excreted in the urine of the people suffering from a disease called multiple myeloma. And what is multiple myeloma? Plasma cell tumor. It has a molecular weight of 45,000. It consists of light chains. What is a structure? It consists of light chains of immunoglobulins, either the kappa chains or the lambda chains. These are synthesized in excess. These chains, they are synthesized in excess, and either of them are excreted in significant amount in the urine. So Ben-Jones protein are actually the kappa or the lambda chains of immunoglobulins, and they are synthesized in excess and excreted in urine. What are the immunoglobulins? Antibodies, which help you to fight against infections, which provide immunity. This we'll study as a separate topic, and then all the confusions will be removed. Now here you can say B lymphocyte, antigen is coming. This is the plasma cell. Immunoglobulins are stimulated production, क्योंकि आपको पता है कि antigen, they stimulate the production of the antibodies, immunoglobulins. And immunoglobulins are composed of lambda chains and the kappa chains. And in the urine, if these are synthesized in large quantity, then they are excreted in urine and they are called Ben-Jones protein. Here you can see the kappa chains, light chains, kappa chains or the lambda chains. Normally, very small quantity of the kappa or the lambda immunoglobulin light chains are excreted in urine. They pass through the, why normally only very small quantity is excreted in urine? Because they pass through the glomerulus filter but are reabsorbed by the kidney tubules, and all the things which are reabsorbed by the kidney tubules, they are not excreted in urine. Okay? So they are reabsorbed by the kidney tubules and broken down in the lining cells and thus not found in the urine. That is, in the tubular cells. In the tubular cells, they are broken down and therefore they are not found in urine. There is a test to identify the Ben-Jones protein, and this is the heat test. On heating the urine 50 to 60 degrees centigrade, Ben-Jones protein are precipitated just like albumin. Okay? But when heated further, they dissolve again. Albumins are not dissolved on heating further, but Ben-Jones protein, they are dissolved when heated further. So what we have learned from this, that Ben-Jones proteins are excreted in the urine of the patient with multiple myeloma, and they are composed of the kappa or the lambda chains. When they are synthesized in extra amount, then they are excreted in urine. Normally, Very small quantity of the kappa or the lambda immunoglobulin end chains are excreted in urine because when they pass through the proximal tubule, distal tubule, they are reabsorbed and they are broken down in the proximal and the distal tubular cells, and thus they are not found in the urine. Cryoglobulin. These are proteins which are coagulated with when plasma or serum is cooled. अभी तक देखा hot पे होता है. Now they are coagulated when it is cooled to very low temperature. Most commonly, they are immunoglobulins IgG, IgM. Don't get confused. We will see all types of immunoglobulins in the last lecture. So mostly they are immunoglobulins, IgG, IgM, or a mixture of two. You only remember that immunoglobulins, they are stimulated in reaction to antigen, and then the antigen antibody reaction takes place, and in this way the antigen is destroyed. Traces are present even in normal individuals, and again I am telling you the molecular weight, but don't confuse with it, and you can see that the molecule is large. They are increased in rheumatoid arthritis, lymphocytic leukemia, multiple myeloma, lymphosarcoma, and all these diseases. Next we come to the fibrinogen. फिब्रिनोजन का तो बहुत क्लॉटिंग मैकेनिज्म देखी होगी आपने in the physiology, and you must be very well knowing the fibrinogen. A soluble glycoprotein, carbohydrate is combined with protein and also called clotting factor 1, as it takes part in the coagulation of blood, and it is the precursor of fibrin because fibrinogen forms a fibrin which is actually the blood clot. Normally consists of 4 to 6% of total proteins in blood. Like globulins, it is precipitated by 1/5 saturation with ammonium sulfate. I have shown you what is 1/5 saturation with ammonium sulfate. And in the lab also, I will demonstrate you how the saturation solutions are prepared. Again the molecular weight, leave this. It is a large asymmetrical elongated molecule having an axial ratio of 20:1. It means length is more, breadth is less, elongated. ठीक है, लंबा है. Being asymmetrical and large, it provides viscosity to the blood. What is the function of viscosity? That is, viscosity actually it offers resistance to the flow, and therefore the blood, it does not passes at once. It moves slowly through the blood vessels. Site of synthesis is liver. So you can see most of the proteins, or you can say all the proteins are synthesized by the liver. Structure. It is made up of 6 polypeptide chains. This you have to remember. 2 alpha chain, 2 beta chain, 2 gamma chain. ठीक है भाई? And these chains are linked by disulfide linkages. The A and B portion of alpha and B beta chain, fibrinopeptide A, right? The chains which are alpha chains, and fibrinopeptide B, the beta chains, right? So they are known as the fibrinopeptide. Fibrinopeptide A, which is composed of A alpha chains, and fibrinopeptide B, which is composed of B beta chains. Here you can see 2 beta chains are present and 2 gamma chains are present, 2 alpha chains are present, and here the disulfide in yellow, the disulfide bond or the linkages which is keeping them together, and here you can see that the molecule is elongated. The NH2 terminal region of 6 chains are held together by disulfide linkages, I have just shown you, whereas the carboxylic terminal region are spread apart, that is, they are far. At the end of the molecules, there is a carboxylic terminal, and this gives rise to a highly elongated molecule. अच्छा, अब यहाँ पे एक बात याद रखने की है जो कि you can get in the MCQ. It also contains an unusual tyrosine O sulfate residue on FPB chain, fibrinopeptide B chain. अच्छा, the amino terminal ends of the chain, they bear excess of negative charges, right? Due to the presence of aspartic acid and glutamic acid. So, ये negative charges, what is the role of these negative charges? The solubility of fibrinogen in plasma, they keep it into the soluble form and prevent the aggregation. क्योंकि similar charges होते हैं, they repel together, and therefore the molecule remain in the elongated form. Alpha-2 macroglobulin. It is a large plasma glycoprotein. Again, glycoprotein, carbohydrate is combined with the protein. It is a tetramer, 4 units. It is made up of 4 identical subunits. And it compromises, or it makes 8 to 10% of the total plasma protein. So 8 to 10% जो plasma protein होते हैं वो क्या होते हैं? Alpha-2 macroglobulin. The protein is synthesized by a variety of cell types: monocyte, hepatocyte, astrocyte. Astrocytes क्या होते हैं? Brain के cells होते हैं. Hepatocytes क्या होते हैं? लिवर के सेल्स होते हैं. मोनोसाइट्स क्या होते हैं? प्लाज्मा होते हैं. These proteins have unique internal cyclic thiol ester bond. This you have to remember. Thiol ester bond, which is formed between cysteine. देखो, cysteine contribute SH group, thiol group, and glutamine, it contributes ester group, right? So in this way, a thiol ester bond is formed between amino acid cysteine and glutamine. This group of plasma protein are designated as thioester plasma protein family. Functions. Alpha-2 macroglobulins, they bind to proteinases and is rapidly cleared them from the plasma, right? They bind the proteinases, harmful enzymes, if not required in the plasma, they bind with them and rapidly clear them from the plasma. Second function, very important function, cytokine delivery to the target tissue. Alpha-2 macroglobulin bind to many cytokine and target them towards the particular tissue or cells. ठीक है? For immune response. देखो, एक काम तो इसका ये होता है कि harmful enzymes से which can denature which can destroy any organ, तो उनको ये clear करते हैं. Bind with them and clear them from plasma. And rapidly clear them from plasma. दूसरा काम क्या करते हैं? Cytokine delivery to the target tissue. Alpha-2 macroglobulin bind to many cytokine and direct them towards a particular tissue जहां उनको अपनी immunity का function करना है. Because you know that cytokines का क्या है? Immunity provide करते हैं. Cell क्या करता है? Cell takes up alpha-2 macroglobulin cytokines complex. इनसाइड द सेल जाते हैं, साइटोकाइंस उससे अलग हो जाता है और अपना फंक्शन करता है. Here we can see protease, right? ये देखो, ये protease घूम रहा है and it is a tetramer. 1, 2, 3, 4. ठीक है? 1, 2, 3, 4, tetramer, right? And this tetramer, it binds with the harmful enzymes, right? Tetramer binds the harmful enzymes, and this way bonded protein is excreted from the system. It removes this from the blood. Transportation of zinc. Approximately 10% of the zinc in the plasma is transported by alpha 2 macroglobulin, and the remainder is transported by the